Welcome to Modern Group of Institution, practical series of Anatomy and Physiology First BP 107P. In this video, we study about compound microscope. That is the first practical of the syllabus, that is to study compound microscope. The contents of the videos are object, requirement, theory, how to use a compound microscope, and application of compound microscope. The object of today's video is to study compound microscope. The requirement for this experiment is compound microscope. Now, the theory, a high power or compound microscope achieve higher level of magnification than a stereo or low power microscope. Stereo means a normal microscope. It is used to view smaller specimens such as cell structures which cannot be seen at lower level of magnifications. Essentially, a compound microscope consists of structural and optical components. Basically, the compound microscope is divided into two parts. First one is structural component. The three basic structural component of microscope are head, base, and arm. The head or body houses the optical part in the upper part of the microscope. The base of the microscope supports the microscope and houses the illuminator. Arm that connect to the base and support the microscope head. It is also used to carry the microscope from one place to another place. The second part of the compound microscope is optical component. There are two optical system in compound microscope. First one is eyepiece lenses, that is the lenses which are nearer to your eye, and second one is objective lenses, the lenses which are at the object place. The first eyepiece or ocular is what you look through at the top of the microscope. Typically, standard eyepieces have a magnifying power of 10x. Optical optional eyepieces varies from 5x to 30x. The second part of the optical component is eyepiece tube. Eyepiece tube hold the eyepiece in place above the objective lens. Binocular microscope head typically incorporate a diopter adjustment ring that allow for the possible inconsistency of eyesight in one or both eye. Whereas the monocular single eye uses microscope does not need a diopter. Diopter means by which you can see with both the eyes. In single monocular, you can see the image with the single eye, one eye is closed. Binocular microscope also have swivel, that is interpupillary adjustment, to allow for different distance between the eye of different individual. Now, objective lenses are the primary optical lenses on a microscope. They range from 4x to 100x and typically include 3, 4 or 5 lenses on most microscope. The objective can be forwarded for rear facing. As you can see, the diagram of the compound microscope, it contains the various parts. As we studied in the second slide, that the microscope, compound microscope is divided into two parts. One part is structural component, another part is the optical component. The structural component is mainly gives the structural outline of the microscope, whereas optical component gives the optical component of the microscope. As you can see, eyepiece, draw tube, body tube, nose piece, objective lenses. Eyepiece is optical, part, draw tube is optical part, body tube is structural part, nose piece, objective piece, these all are again optical part, stage, slide, clips, diaphragm, filter, condenser, concave mirror, these all are optical part, whereas foot is structural part. And this portion, as you called as limb, it is also an structural portion. Automatic stop, fine adjustment and core adjustment are also part of optical component. Now the another optical component is nose piece, houses the objective. The objective are exposed and are mounted on the rotating turret so that different objectives can be conveniently selected. The standard objective includes 4x, 10x, 40x and 100x, although different power objective are also available for the compound microscope. Next one is coarse and fine focus tube are used to focus the microscope, are used to focus the object. Increasingly, they are coaxial knobs. That is to say, they are built on the same axis with the fine focus knob on the outside. Coaxial focus knob are more convenient since they viewer does not to grope for a different knob. Next part of the microscope is stage, is where the specimen or the object to be viewed is placed. A mechanical stage is used when working at higher magnification with delicate movement of the specimen slide are required. Next part of the optical compound microscope is stage clips. Stage clips are used when there is no mechanical stage. The viewer is required to move the slide manually to view different section of the specimen. Another part is aperture, is a hole in the stage through which the base light reaches to the stage or onto the object. Next one is illuminator. Illuminator is the light source for the microscope, to typically located in the base of the microscope. Most light microscope use low voltage halogen bulb with continuous variable lightning control located with the base. And in some of the microscope, only a simple mirror, simple concave mirror is used to produce illuminating light. The next part of the compound microscope is condenser. Condenser is used to collect and focus the light from the illuminator onto the specimen. It is located under the stage, often in conjunction with iris diaphragm. Now, last one is, that is iris diaphragm. Iris diaphragm control the amount of light reaches the specimen, because if higher amount of light reaches to the specimen is invisible, and if the lower amount of light reaches to the iris diaphragm, then the specimen is not clearly visible. So iris diaphragm is responsible for the control of light reaches to the specimen. It is located above the condenser and below the stage. Most high quality microscope include an abyss condenser with an iris diaphragm. Combined, they control both the focus and quantity of light applied to the specimen. Now, what is condenser focus knob? The condenser focus knob moves the condenser up or down to control the lightning focus on the specimen. Now, how to use a compound microscope? So first of all, we have to familiarize with the compound microscope. First, familiarize with your microscope, with all the part of the microscope, so that you can easily move from one part to another during operation. Now, moving and placement. It is worth remembering that a good quality microscope will last for a lifetime. It is a sensitive scientific instrument that will suffer damage from sharp blow or impact. Always, therefore, carry your microscope with your both hands. Grasp the arm with one hand and place the other hand the base for support. Always place the microscope on a level and stable surface. Now, the next is slide preparation. Microscope slides should always be prepared with cover slip or cover glass over the specimen. This will protect objective lens if they touch the slide. To hold the slide on the stage, fasten it with the stage clip. You can push down on the back end of the clip to open it. Now, the main important part in microscope is focusing the microscope. First of all, you have to start by turning and revolving the nose piece so that the lowest power objective lens is clicked into position. The lowest power objective is the shortest one. This objective is the easiest to focus and center the image in the field of view. Now, while looking at the objective lenses, and stage from the side, turn the coarse focus knob so that the stage moves upward toward the objective. Move it as far as it will go without touching the slide. Next, look through the eyepiece and adjust the illuminator for light and diaphragm until the maximum comfortable level of light is available. Slowly turn the course adjustment so that the stage moves down all the way away from the slide. Continue until the images comes into broad focus. This is very important. Turn the fine adjustment knob as necessary for perfect focus. Move the microscope slide until the image is in the center of the field of view. Then readjust the illuminator or diaphragm in order to attain the clearest image. Once you attended a clear image, you should be able to change the high power objective lenses with only minimum use of focusing adjustment. If you cannot focus on your specimen, repeat the above step and work from objective to objective until the high power objective lens is in place. Now, what are the applications of compound microscope? Optical microscopy is used extensively for microelectronics, nanophysics, biotechnology, pharmaceutics research, mineralogy, and microbiology. Optical microscopy is also used for medical diagnosis, the field being termed as histopathology when dealing with tissue or smear test on free cell or tissue fragments. In industrial use, binocular microscope are very common. Aside from application needing true depth perception, the use of dual eyepiece reduces the eye strain associated with long workday at the microscopy station. In certain application, long working distance and long focus microscope are also beneficial. So this is all about the compound microscope. Thank you very much from Modern Group of Institution, Modern Institute of Pharmaceutical Sciences, Indore.